what I want to talk a little bit about today deals with motion perception. So we're in chapter 8, and so what we're going to be dealing with. I want to talk first about the neural foundations, because some of the research that's interesting about this comes from Bill Newsom, and it's really it's a good example of them really using the empirical method to examine these topics. And then we'll end this particular lecture with an activity. So let's start here, and what we know, and this seems to be my favorite figure, but it's so clear and easy to use. We know that in primates there is this well-defined pathway setup, and we've kind of talked about this, the what and the where, or the where and the how. But there's actually that well-defined division of the visual pathways that are specialized for motion. And if you follow on up, you start on the magnocellular layers of the LGA, and you move through the striate cortex, and you start to move up on into the dorsal stream. You end up in this best defined area. It's an extrastriate area known as MT, or sometimes you'll see it as V5. And if we actually look at it in an actual figure here, so we're looking right here at area MT V5, and you can kind of see it here. It moves on up into the dorsal stream. But it's different than other extrastriate areas in that almost all neurons in MT are direction selective, almost all of them. Which means, if you remember, they typically respond best to motion within a given range of directions, and respond essentially not at all to motion in the opposite direction. So you saw some of this with the Hubel-Wiesel work when they were discovering the complex cortical cells, but not quite as overwhelmingly direction selective as you see in area MT. Now, behaviorally, these neurons have been linked to analysis of motion of complex patterns, detection of target motion relative to background, as well as generation of signals for smooth eye pursuits. So clearly it's got some more connections than just right there in area MT. Now, enter the work of Bill Newsom. So you're going to see the citations down at the bottom here. They designed a special stimulus that capitalized on this idea of coherence. And this was a really neat method. And the idea is they've got these dot stimuli that they either move together not at all, or on a scale of low coherence to high coherence, some of them move together, or the highest level of coherence, they're all moving in the same direction. So on the left here, you see there's no coherence. The dots are just moving randomly, and there's no overlap in the direction that they're all moving together. Here in the middle, you've got the 50% coherence. Half of them are moving in the same direction. And then the 100% coherence, where they are all moving in the same direction. And so they trained the subjects on this, monkeys specifically, and they were, and this was extensive training where the monkeys had to learn the task, and it wasn't an overnight type of thing. It was an extensive training period so that they knew how to respond given certain tasks, assuming they could detect it. And so what we know is that under pretty favorable conditions, the human and the monkey can reliably report the direction of motion of the coherent signal when the correlation is as low as 3%. Between 3 and 5% is kind of the typical range when favorable conditions are met. Now, they're going to need these correlations to be higher for stimuli that are moving faster or moving slower than the optimal value. And that optimal value, just to kind of give you an idea, it's what they call an interdot interval of 50 milliseconds. The dots are spaced about 1/10 of a degree from one another, and essentially the overall speed is about 2 degrees per second. And so that's kind of the optimal if we're talking about being above that or below that value. Typically need to have a little bit more coherence as you move away from that optimal value. Now what they did was Newsom and his colleagues over multiple years, really starting in the late 80s, they explored the idea that psychophysical performance using this stimulus might actually depend on neurons like those in area MT, which if you make that jump, remember that the MT neurons are direction selective, and we're talking about direction of motion. And we also know that we have this summation idea in these particular neurons, very similarly to what we talked about with Hubel and Wiesel. These neurons can sum visual signals over large areas of the visual field. And so this coherence idea, the more coherence, the more summation might be happening. So they had a pretty good idea that these neurons would be involved. And so it led to quite a few studies. So let me talk a little bit about this late 80s study. And essentially they were asking this: are MT neurons necessary for monkeys to detect motion in the coherence stimulus? And now remember, we're talking about monkeys who've been extensively trained on this task. And so what they did was they studied the effects of lesions of MT using very targeted injections of acid to create small regions of damage in area MT. And so if I pull up the plots from this, just to kind of get you oriented, here on the x-axis of both of these, you've got the speed, and that's the degrees of change per second. On the y, you've got the correlation, and essentially this is the coherence. The higher the correlation, the greater the coherence. And essentially what you can see here on the left is the lesion hemifield. Right? So they only injected to destroy processing in one part of the hemispherical visual field, and then they left the other hemisphere intact, or the hemifield intact. And so you've got the top line, so the solid line here where it says with the circles is pre-lesion and then post-lesion. Okay? And that's true in the other plot as well, pre-post. And so what you can see from these plots is that direction discrimination thresholds in the lesion hemifield were much higher, okay? Meaning that the correlation of dots needed to be much higher in order for the direction of motion to be detected. If you look over in the other hemifield, the one that was unaffected, did not show such a threshold increase. So when I'm talking about, here's the lesion hemifield after the lesion, you can see that the correlation needed to be much higher than at baseline, pre-lesion. But in the control condition, there was no difference. So they used this as a way to show that those MT neurons are implicated in motion detection for this coherence task. Also, it's kind of neat to note that these lesions were small enough that most of these organisms showed pretty full recovery afterwards. The larger the lesion, the less complete the recovery. But for the most part, the monkeys were fine. Now, let me talk a little bit more about Newsom as they explored this more. And I want you to kind of think about this. This is neat. So Newsom and colleagues really were the first to successfully measure behavioral and neurophysiological concurrently, which they actually, and your book makes a nice point of this, and if you ever get online and look at some of the talks that Bill Newsom gives, he's not real shy about saying that his lab was the first one to do this. But it was nice because they actually clearly linked the behavioral and the physiological. So they were actually measuring these MT neurons while the monkeys were actually having to make a response that they had learned. Now that's different if you remember from Hubel and Wiesel. They just had those cats being measured and just being bombarded with stimuli. They weren't having to do anything except look at the stimuli. Here you had monkeys having to make a choice while their MT neurons were being measured. So that's pretty neat. And since they were able to do this, this kind of goes back to some of the stuff we've already talked about when we talked about plots of psychometric functions and things like that. Well, what they did and what they found is that information contained in the MT neurons was sufficient for performance on the motion discrimination task. And so what they did, and you can kind of see it in this plot, is here's your percent correlation and here's the proportion of correct responses on the y-axis. The dark line is what's called the neurometric function, and essentially what the neurometric function is this is the theoretical neuronal choice. It's a probability, it's an estimate to stimulus strength for a particular MT neuron. Okay, so this is basically the idea, given this neuronal response, we should be able to predict that the higher the response, the better the proportion of correct responses in detecting the motion. And what they did is they compared it to the actual psychometric function, so the actual monkey's choice performance. And you can kind of see that if you look at how these two functions are overlaid, they're nearly identical, but I mean clearly they're not. But statistically, they're not different. And so you can kind of see from this that neuronal response is really sufficient for predicting how well they're going to do on this motion task. And in the grand scheme of things, that's what links those MT neurons to the performance and showing that they are truly linked to performance on that particular task. And so, figuring into this, monkey performance should be altered, if you go by this, by activating, artificially of course, MT neurons during the task. And so your book covers this a little bit, but what they did, it's nice that you've got such a very clear organization in your visual cortex, but they found that using little microstimulators to activate neurons that are actually selective for a different, for instance, a different direction of motion while they're being presented with another direction of motion, they actually found that they could shift the response to match the artificial activation. So they found a significant separation between neurometric and psychometric functions. So if you look at a few of these plots, I just put a few. So here again, you've got your correlation, and here's the proportion of decisions that were preferred. And so here, the dashed line is where there's no stimulation. Okay, and when you actually artificially stimulate another neuron, another set of neurons that are actually picking up on a different direction, okay, you actually see the separation between the psychometric and the neurometric kind of function here. So you've got what they're doing while they are not being artificially stimulated and everything's all good, but then when they're stimulated you see a shift in their response. Same thing in this plot. You see a shift in their response when you artificially stimulate another set of neurons in MT. What that means is it's just another step that Newsom and colleagues did to show that MT is linked to performance of this motion detection. So if you have a set of neurons, for instance, that are direction selective for right, and you see them doing just fine when the stimulus is going in the direction that is right and matching those neurons, but if I artificially activate the direction selective neurons that are for left while I'm seeing a stimulus that's going right, we can actually shift their behavior to select left even though the stimulus is right. So again, implicating these MT neurons in performance on this task. Now here's the difference. Remember, this is all kind of a static situation, and remember that a lot of this is actually done in really sterile laboratories. The monkey and the organism or the subject or participant is seated, and there's no movement. The movement is actually happening in the stimulus. So what I would like you to do, if you're in the Monday, Wednesday class, we actually had a chance to do this in class. If you're in the Tuesday, Thursday class, you need to answer this question and email this to me so I can give each person feedback on this. So here's the activity. The research I just covered, like I said, is from a static situation, but what we know, especially from the work of Gibson and the ecological approach, is that in most cases, when we're perceiving things, it's occurring in a dynamic situation. Right? We're not always still. We're always behaving, and there's this reciprocal relationship between perception and action. So what I'd like for you to do, and you can email this to me, it's not a bad thing for either class to email this to me, of course, because it is one of your essays, hint, hint, wink, wink, nudge, nudge. Can you explain the theory on how we perceive motion in a more dynamic sense? So in other words, how might we perceive motion when we are moving? And if you answer that with that particular theoretical perspective, make sure that you can discuss the evidence that seems to support that theory.